Okay, so I'm going to talk to you about multimodal static and dynamic connectomes. The first part of the talk will be just looking at static connectomes and looking at one of the issues that has come up in a bunch of groups really, which is looking at the issue of individual partial variability. And then I'll go on to look at how we can start to compute connectomes using electrophysiological data in a very similar way to what we can do with fMRI. And then in the second part of the talk, I'm going to talk about using techniques that are potentially solving some of the problems that we have with sliding window approaches to move towards, hopefully, the kinds of dynamic brain network analyses that even Steve Peterson will believe in, but we'll see. So over the last several years or so, there's been a lot of work done really looking at trying to relate features from functional connectomes to behavioral data, and we saw some nice examples of that in Emily Finn's talk. This is one example here, which was published recently, which basically used canonical correlation analysis to relate multivariate features from behavioral data to functional connectome edge strengths. And they basically found a CCA mode that picked out behavioral features that related to positive behavioral traits and negative behavioral traits as being distinct from each other. And this was shown to be significantly correlated with a bunch of edges in the functional connectome, a CCA mode, which picked out edges associated with the default mode network. So these kinds of analyses are becoming increasingly used and increasingly important to give us insights into how large-scale networks interact with behavior. But there is one big kind of issue, which, amongst many others, of course, that have been discussed already, but one that I want to highlight now is this issue of between-subject variability. And this is just showing you, for the sake of example, the way in which the language network shows a huge amount of variability over different subjects. Just a movie showing you the language network for a bunch of different subjects. And you can see the different nodes of the language network really moving around in space really quite considerably. And if you were to use this as a spatial basis function, and you'd see similar things with parcellation approaches, as your way of extracting time series for your functional connectivity analysis, then these variations are going to leak into your functional connectivity calculations unless you do something to take them into account. In other words, individual variability in spatial maps and parcellations will translate or can translate into apparent changes in functional connectivity. So what can we do about this? Well, a typical approach is to take a two-step procedure whereby you first of all define your group atlas. And then on the second stage, you would then take that as a fixed atlas and look to estimate how individuals vary around that group atlas, using something like dual regression, for example. An alternative, which is an approach that we prefer, is one where you essentially do this all in one, where you combine the estimation of the group atlas with your estimation of how individuals vary from that group atlas. And that's got the benefits of allowing you to regularize in both directions. So the estimate of the group atlas will benefit from the estimates of the uncertainty and variance at the individual subject level. So the approach that we have for doing this is being developed by a postdoc in our lab, Sam Harrison. It's published about a year or so ago, called Profumo, which stands for Probabilistic Functional Modes. And the model is essentially the same kind of model as you see in an ICA model, an outer product model, that models the spatiotemporal data for each subject as a linear combination of modes, where each mode is described by a spatial map and by a time course. And this is described at the subject level, including a model of the functional connectome, telling you the mode-by-mode correlations between the different mode time courses. And then we have the group model, which represents the mean and the variance around the spatial maps, and the same for the connectomes, which is top-down regularizing what's happening at the subject level. And we can infer on this model all in one using Bayesian approaches. So this will spit out group average descriptions of your functional connectome, as well as individual subject descriptions. But also, importantly, it will spit out group averaged spatial maps, or spatial basis sets that are being used to compute the connectome, as well as individual subject versions, which tell you about the variability of that subject's spatial maps around the group average. So what can we do with this information? Well, of course, what we should be doing is looking to revisit these kinds of analyses where people have related functional connectomes to behavioral data. and basically allow ourselves to consider the possibility that the variability of the subjects can be described not just in functional connectomes, but also in these spatial maps. And to sort of cut to the chase, and in the interest of time, what we seem to be finding when we do this is that actually what previously has been assigned as being a functional connectivity difference that's correlated with behavior is actually instead caused by variability in the spatial maps. In other words, the variability in the location of functional regions. And if you want to find out more about this, then I encourage you to go and see these two posters later in the week, one by Sam Harrison and one by Janine Visterbosch. So we've seen a lot of work about using fMRI to compute connectomes, and that's obviously very nice in terms of the rich space information that you can obtain. But of course, you're always looking at what's happening through the blurred lens of the hemodynamic response function. So we're missing out on what's happening at much faster timescales, the kind of timescales you'd associate with fast cognition. And so one alternative is to use electrophysiological techniques like MEG and EEG, which are also non-invasive, to get some insight into what's happening at these fast timescales. So in the last few years, there's been some nice work done in the field of electrophysiology to show that you can establish a nice pipeline for computing functional connectivity. And the kind of technique that's been developed is working very well on resting state data has been one that uses correlations not on raw time courses like you do in fMRI data, but correlations on amplitude time courses. So the pipeline that you typically use is, as you can see here. I won't go into the details. The main point is when you get through the pre-processing, you compute correlations on amplitude time courses rather than on raw time courses like you would do in fMRI. And really the insight as to why you do this is that MEG, like EEG as well, is a direct neuronal measure. And so you're exposed to the fact that you now have potentially non-zero lag conduction delays between different brain areas. And dealing with raw time courses, you've got to somehow take that into account, which is non-trivial. Whereas if you work with amplitude or envelope time courses, you basically make yourself sort of resistant to the non-zero lag conductances. So people have shown when you do this, you can build up nice functional connectivities in MEG data that kind of what you'd expect, given what we know from fMRI data. So functional connectivity between bilateral areas of primary sensory and motor systems. And you get one new insight from this approach, which is that the carrier frequencies of the oscillations, which you find sensible functional connectivity, tend to be in the low frequency ranges. So we're looking at oscillations in the alpha and the beta band between 8 and 30 hertz. So we have used this recently, along with fMRI data, to look at the question of heritability of connectomes, using data from the Human Connectome project. And the HCP has the benefit of being multimodal, but also having a twin structure in it, whereby some of the pairs of subjects are monozygotic twins, and some of the pairs are dizygotic twins. So what we can do is use the extent to which there's shared genetic material to see how well that predicts the similarity in the functional connectomes. So first of all, we can look at MEG data. Here we're focusing on the alpha band, and we can see here the connectome we compute using a 38 cortical region parcellation and amplitude correlations of the sort I've been describing earlier. And this computes a nice looking densely interconnected occipital network with dorsal and ventral streams in the kind of way you'd expect for an alpha signature in electrophys data. And then we can look to see the extent to which the shared genetics predicts the effects of or the similarity in your connectomes, and we can find a nice significant effect when we look at that. But we can go further than that and actually look at fitting a linear variance components model and show that the effects of shared genetics significantly outweighs the effects of shared environment. And we can also look at the fMRI data in the HCP, of which there's a lot more data, approximately 10 times as much, and we see the same effects. We see the same effects of heritability, and we see the same effect of shared genetics outweighing the effects of shared environment. And the effects are even stronger, mainly because of the huge amounts of extra data you have in the fMRI data. So, I mean, one reason for having this kind of analysis, apart from looking at heritability, is really showing that we can now do things in electrophys which are very similar to what we can do in fMRI data. Okay, so I'm going to talk now about how we might go about computing dynamic connectomes, picking up hopefully from where Steve Peterson left off. And he talked about using sliding window approaches. And these are pretty straightforward, as was described. You rather compute your connectomes on the entire scanning session's worth of data. You instead compute your connectome on a limited portion of data. And then you can slide that window through time and build up a time course of connectomes. And when you combine that with clustering techniques, people have proposed that you can find evidence of temporal non-stationarity, although of course, as Steve Peterson has talked about, there is concerns about the actual validity of those results. People have also looked at sliding window approaches in electrophysiology as well, where they, for example, have found periods in time of particularly heightened functional connectivity and shown if they focus their analyses on these windows in time, that they can better identify resting state networks in MEG than if you use a temporarily stationary approach. So one of the big problems with sliding windows really comes down to the question of how you go about choosing the width of your window. If you make that window too short, then you'll get unreliable estimation. Of course, you can try and correct for that with sensible statistics, but you're going to lose sensitivity. Or if you make your window too long to try and solve this problem, you may well be missing genuine quick changes in your data, if they are indeed there. So even with MEG data, we tend to end up using windows on the order of several seconds in width. And in fMRI, those people that have tried to do this end up using windows in the order of about a minute. So we're really missing out on potentially interesting information at fast timescales, particularly in the electrophysiological data where we have like millisecond temporal resolution. So rather than using sliding window approaches, one approach that we've been developing is to think about how we might break up that sliding window into a bunch of potentially much shorter lived visits to some idea of a repeating brain state, whereby each of these individual visits to a brain state is providing far too little information, far too little data to allow you to estimate your connectome. But if you can simultaneously identify when you come back to the same brain state, then you can pool over all of these visits to get a good estimate of your connectome. And then you can imagine that might be brain state 1, and at other points in time you visit other brain states characterized by their own distinct functional connectomes. So this description of the ongoing activity in task or in rest as being made up of a sequence of brain states where each state is characterized by a distinct connectome is what we can actually compute using an approach called hidden Markov modeling. So the generative model for a hidden Markov model is pretty much as I described on the previous slide. At the core of the model, you have a bunch of state time courses, which are basically binary state time courses telling you about how the states switch on and off. We make the important assumption that the brain states are mutually exclusive, so only in one particular brain state at any one point in time. And then we have the observation model, which says if I know what brain state I'm in, what can I predict about the neuroimaging data I'm going to record in that brain state? And for this, we use a very simple multivariate normal distribution, at the core of which is a covariance matrix with a brain area by brain area correlations that correspond to our connectome. And of course, you could do things that Gail Varacro talked about and invert those to get partial correlations if you wanted to. But the key point here is that we have a different connectome, a different correlation matrix for each of our brain states. Okay, so this approach is like a general framework. You can apply it to a wide variety of different multimodal data sets, either separately or simultaneously. I haven't got time to talk to you about the applications to intracranial recordings, looking at LFP data and also spike data. And we also have applications in EEG and simultaneous EEG and fMRI data. I'm going to focus now on fMRI and MEG and start with the fMRI data. So we're going to see if we can infer dynamic networks in resting fMRI using the HMM and look at the nature of the dynamics that we see. So we're going to use the data from the Human Connectome project. We've got approximately 1,000 subjects, 1 hour each, so it's a huge amount of data, 1,000 hours worth of data. And so we need to use techniques which can cope with big data or obese data, as Victor prefers to call it. And for this, we use a variant of HMM that can cope with this huge amount of data based on stochastic learning. It basically iterates between subsets of the data, subsets of subjects of data, and then infers on the whole group model at the same time. It's worth pointing out that when we do this with, let's say, on the order of 10 states, we use 12 in practice, but for the sake of argument, let's say we have 10 states, we've essentially got 100 hours worth of data to estimate each state's functional connectome when we take this approach. So this is really the motivation, or the reason why we're putting this forward as a very nice way of overcoming the problems with sliding window approaches. It's a huge amount of data that's being inferred, being used to infer each of the state's functional connectomes. So the pipeline we have is, as you can see here, you pre-process your fMRI data. We get voxel time courses. We can then compute specific parcellation time courses or component time courses using dual regression on, in this case, a 50-component group spatial ICA basis set. And then on this data, we can compute the hidden Markov model to get a bunch of state time courses. And we also get activity and connectomes separately for each of the brain states. So here I'm showing you the activity maps for 6 of the brain states. So this is telling you in a particular brain state which brain areas are active compared to what's happening on average. And you see the same kind of combinations of resting state networks you see in other approaches like ICA, so a combination of cognitive and primary sensory and motor states. What's happening temporally? Well, we get a very nice mixing of the state time courses. I'm showing you just 3 of the state time courses here in a short block of data. And we can summarize the state switching using, for example, the state lifetimes. So this is the amount of time you spend in a state before you move into a new state. And the switching happens on the kind of 5 to 10 second time scale, which is really quite fast, but it's kind of suggesting here that we can find switching on time scales as fast as you can go given the hemodynamic limitations that you have in fMRI data. So given this, we want to then go and have a look at the state time courses and see if there was temporal structure in the state switching at slower time scales. So first of all, we looked at the kind of simplest measure we could think of, which was the amount of time that each subject spends in a particular brain state. This is quite simple to compute for each subject. Here for the 12 states, the proportion of time that you spend in each of the 12 states. And then you can repeat that for all subjects. And then we take this and compute the correlation matrix over subjects to get this states-by-states correlation matrix. And this has got a really amazingly striking structure in it, whereby it partitions into 2 groups of states, one of 4 states and one of 7 states for our 12-state HMM, with state 5 not really being belonging to either of these two groupings. And you can do hierarchical clustering to confirm what you can see by eye, whereby this data falls into these 2 groupings that we call metastate 1 and metastate 2. So what do these metastates correspond to? Well, if we look at the connectomes summarized in the form of degree maps, we can see that metastate 1 corresponds to primary sensory and motor brain states or networks, and metastate 2 corresponds to cognitive brain states or networks. So what we're essentially saying is if you know how much time a particular subject spends in the default mode network state, then you can predict very highly how much they would spend in other cognitive states, say the salience network. And the same is true for the primary sensory and motor states as well. We next looked at the information in the sequencing of states, in the sort of simplest sequence you can think of, which is a pair of brain states. So we looked at one of the things that's actually inferred as part of the hidden Markov modeling via its Markovian assumptions, which is the probability of moving from one state to another state. And what you can see here is it's not quite as striking as the correlation structure in the occupancies per subject, but if you look at the plotting of this matrix in a different form and do statistical testing, you can show that there's, again, these two groupings of states, whereby you tend to cycle between the states that correspond to primary sensory and motor states, and correspondingly tend to cycle between states that correspond to cognitive states, with much more infrequent transitions between those 2 groupings. So if you want to find out more about this, there's a whole bunch of stuff also showing how this relates nicely to behavior and genetics, and some demonstrations of the reliability and robustness of these results. Then please go and see Tigo Fedore's talk later in the week on Wednesday morning. So I'm just going to finish off by looking at using the HMM on the electrophysiological data, MEG data, where we potentially have much richer temporal detail to tap into to really benefit from these dynamic techniques. So again, we're going to start with resting state. and we're going to see how fast the dynamics are. The pipeline that we have is very similar to what you saw for fMRI data, but rather than looking at running the HMM on the raw time courses, we run it on the amplitude time courses for the reasons we talked about earlier when we were computing static connectomes. And again, we can spit out the state time courses and maps of the relative activity over states and the connectomes. So in this first approach, we were basically able to show that we could compute spatial maps that correspond to the typical resting state networks that you see using other approaches. But what was particularly striking here was that the dynamics were really very, very fast, as we were hoping they would be. So the lifetimes here, which show you the amount of time you spend in a brain state before you move into a new brain state, on the order of 100 milliseconds. So here you're seeing these resting state networks switching or showing dynamics on timescales, sort of orders of magnitude than have been shown previously using techniques like ICA. So that was kind of nice, but we were kind of frustrated by the fact we had to work with amplitude time courses. And so we wanted to see if we could get this working on meg data in terms of the raw meg time courses. This could allow us to tap into faster dynamics, but also has the attraction that we can look for a very much more specific mechanism of neuronal communication in terms of phase coupling, in support of the idea of communication through coherence that's been put forward by people like Pascal Fries. So the way we can do this is by running HMM on the raw time courses, but not in isolation, but by running it with lagged versions of each of the brain region's time courses. So you take brain region 1, include its raw time course, then you include also the same time course lagged by tau, and then by 2 tau, and then by 3 tau, and so on. And you can repeat that for all brain regions. And then what the HMM can go and do is you can go in and find where there are correlations between different lagged versions of different brain region time courses, thereby finding non-zero lag interactions, which allows you to find not only amplitude coupling, but also this phase coupling that we're particularly interested in. So we're going to run this on resting state data using a 38-node parcellation on the raw time courses, focusing on the low frequency ranges that have been successful in connectivity and MEG in the past. So we compute the relative amplitude maps first of all, and you can see that we get the same mix of cognitive and primary sensory and motor networks that we are used to seeing, broadly speaking. But the question is, can we find states that show transient events of distinct phase coupling? And if we look at these, then indeed we do. So here I'm showing you the coherence, which is the measure of phase coupling between 1 and 30 hertz, showing you the connectomes specifically for the 8 different brain states. So you can see very nicely that the visual state, for example, has phase coupling between visual areas, whereas the motor network has phase coupling between areas of the motor cortex. So one particularly intriguing result from this is that, unlike other approaches, what we find here is a splitting of the default mode network into 2 different subdivisions: an anterior default mode network with phase coupling between frontal and medial prefrontal cortex, and also medial temporal lobe areas. And then very distinctly, a separate putative posterior default mode network with phase coupling between parietal areas and the PCC. So we can go further than this and look at these 2 networks in a bit more detail and look at the specific frequency information that exists in them. Here, this top line you've already seen, the wideband perspective, but we can look at it separately for delta, alpha, and beta bands as well. And now you can see that the anterior default mode network is really doing most of its stuff in the delta band. So there's really strong, nice delta-specific phase locking between the frontal areas and the medial prefrontal cortex and medial temporal lobes. But also quite intriguingly is this phase coupling between frontal areas and the PCC. And by contrast, the putative posterior default mode network is doing all of its stuff very differently in the alpha band, with the phase coupling with the PCC. So what we have here, I think, is the reason why we see these 2 subdivisions of the default mode network. Essentially, they're doing their communication in very different oscillatory regimes at different timescales, and possibly reflecting the different timescales at which these 2 subdivisions of the default mode network might be operating cognitively. But we need to do future work to confirm that. So to summarize, we looked at static connectomes and saw that we can now compute them quite nicely in electrophysiology as well. And we discussed the issue of spatial misalignments, which is something I think increasingly we need to be really careful about in how we interpret functional connectivity and its relationship to behavior. And then we looked at this idea of potentially using hidden Markov modeling to overcome some of the problems that we have with sliding window approaches. And we looked at applications in fMRI and also in MEG. And just finish by saying thank you to all the collaborators involved in this work, and thanks for listening.